Our research proposal focused on the use of senolytic treatments in mice. Specifically, we researched how a senolytic combination of dasatinib and quercetin can regenerate heart muscle tissue in mice infected with myocarditis.
For some background, senescent cells resist apoptosis and do not undergo cell divisions. According to the accumulation of senescent cells theory of aging, the accumulation of senescent cells in tissues can cause chronic diseases and inflammation by releasing toxic chemicals such as senescence-associated beta-galactosidase positive, which we will refer to as beta-gal. The accumulation of beta-gal causes many of the inflammatory diseases of the heart muscle tissue. Inflammation in the myocardial tissue can be recognized as a leading cause of heart disease. Since myocarditis can be understood as an inflammatory disease of the heart muscle tissue, looking at this figure, you can see how inducing this disease using the CVB3 virus model in mice can allow us to study how to decrease inflammatory heart disease. Our intervention will be a senolytic drug cocktail to be administered to lab mice consisting of 5 milligrams of the dasatinib and 50 milligrams of quercetin. This treatment will be administered using an oral vehicle in order to target and kill senescent cells that are accumulating the B-gal. Looking at this figure, you can see how the treatment should hopefully target the inflammatory compounds, and the systemic deletion of cells with high levels of B-gal in the mice should reduce inflammation in their heart muscle tissue, as well as increasing the rate at which their heart tissue cells regenerate. We chose mice for our experiments because they are inexpensive and genetically similar to humans in the way that they develop diseases. Additionally, they have a complete genome map and the ability for genetic modification. Specifically, we looked at wild-type C57BL6, as the effects of the interventions performed are already understood in this breed. We will have a randomly assigned control group and treatment group, both infected with myocarditis. We will separate the groups by gender and age, as well as taking notes of their BMI throughout the study. Half of the group will need to be sacrificed for the beta-galactosidase assay, and the other half will be used for the longevity studies. After the intervention, we will perform a series of tests on the mice: grip strength with a grip strength meter, a balance beam walking assay, a treadmill running endurance test, blood glucose tests, RNA sequencing, and a B-gal level assay. The anticipated results of our D plus Q treatment would be an increase in the regeneration rate of myocardial heart tissue, a decreased rate of extrinsic aging, and a greater longevity compared to the control mice. The reduced amount of B-gal in the myocardial mice heart cells should result in a reduction in the development of age-related heart diseases and in an improvement in health span and physical fitness.
So here are some promising studies that we are basing our proposal on. This first study demonstrates that our drug has already shown improved physical function and increased lifespan in old age. The second study shows that D plus Q has decreased coronavirus-related mortality over time in old mice. What we are essentially doing is combining two studies that have already shown promise in old mice. Our study is unique that it specifically looks at myocarditis.
This research proposal is also especially significant as myocarditis has become increasingly prominent health concern with the onset of the COVID-19 pandemic. Viruses like the one that causes COVID-19 can induce myocarditis in individuals of all ages.
Heart disease is typically an aging-related disease, but myocarditis impacts both old and young populations. While we are confident that we will see improved function in old mice, there is a possibility that this will extend to younger populations in our study.
For human applications, it may be valuable to examine whether COVID-19 induces myocarditis in mice by a similar mechanism to how it does in humans, because this could enable the use of D plus Q to relieve symptoms and adverse effects of COVID-19.